organisms. They are unicellular organisms. And their size is ranging from what? 0.2 to 2 micrometers. Their size is 0.2 to 2 micrometers, and they are microscopic. They are not able to see with your naked eye. So if you wanted to see the microorganisms or bacteria specifically, you should use the microscopes. And we have studied about different types of microscopes. Under that you have studied about light microscope, electron microscopes, and so on, right? So if you see these bacteria, they are different in shapes and sizes, right? That's why they are called as pleomorphic. What it is? Pleomorphic. Pleomorphic, and they are present everywhere in different habitats. Habitats means the place where they live. Okay? They are present in the soil, they are present in the water, they are present in the air. Right? So here, based on their habitat and different structures, they are having varied morphology. You call it as morphology, right? So when you see there, we have studied already the sizes. When you see, they are present in cocci forms, right? As you can just say, an average size of it, right? And generally, you can see these about RBCs are also given, right? And when you take the typical eukaryotic cell, this is nearly, its volume is 0.3:1, that is, right? It is higher than the bacteria, you can just say. If you see here, these are the different shapes of bacteria. What are those? You can just see cocci forms, cocobacillus. That means they are similar, like a combination of bacillary forms and cocci, and it's not round or it's not rod shape. So that's bacteria are called as cocobacillus. And next one is Vibrio, comma shaped bacteria. If the bacteria are present as comma shape, their shape is as comma, you call it as Vibrio. Best example is Vibrio cholera. Cholera causing bacteria. Cholera disease tells you it's a waterborne infection where it infects the gastrointestinal tract, right? So that bacteria is in the form of a comma. That's why it is called as Vibrio. Andukane daniki peru em icharata? Vibrio cholera. The shape is in the comma shape, or the structure of the cell is in comma shape. That's why it is called as Vibrio cholera. And next one, you can just see Bacillus. Bacillus form. Bacillus form means it is in the rod shaped bacteria. A rod shaped bacteria is called as Bacillus. And next one here you can just see Spirillum. Spirillum means spiral shaped bacteria. They are in the form of a spiral, like spring. Okay, that's why they are called as Spirillum. And next one is Spirochetes. These are the specific group of bacteria. You can just see plague disease and winner kada. Plague, or you can see syphilis. Sexually transmitted disease, it is syphilis. It is caused by spirochetes, spirillum, right? It is spirochete which causes syphilis. Palladium, it is bacteria which causes syphilis. This is a specific group of bacteria. Only specific cells will be appear in the form of spirochete. So these are the different shapes of bacteria. You can just see what are the different shapes, right? What are those? Cocci forms, what I told you, they are in the form of circular or round shaped bacteria. Those are called as cocci forms, and they may be arranged as a single form or sometimes in the form of a pair. We have even studied this one, right? Single, in the form of a single circular cocci, it is called as monococcus. If it is in the form of a pair, joint we call it as diplococcus, and if you call it as tetrad. This is because, as I told you, during the division, if the division doesn't happen properly, right, complete division jaragadu. Okay, complete division jaragadu, rendu cells separate ga ayipotayi. But kani konnisaarlu em jarugutundante, you may not have a perfect division. Sometimes that here binary fission. So that time there may be joints, okay, between the two cells. That is improper division. Okay, and here I just told you, sometimes they may be in the form of a chain laga. right? That you call it as streptococcus in the form of a chain. Chain madriga continuously cocci forms are arranged one after the other. These are called as streptococcus. Cocci forms which are arranged in the form of a chain. So those are called as streptococcus, right? And staphylococcus are also there. Staphylococcus means if they are arranged in the form of a bunch, great bunch-like structures. If they are present in the form of a great bunch, right? So these are called as staphylococcus. Generally, the streptococcus and staphylococcus bacteria are present on our body. ఇప్పుడు మన బాడీ మీద నుంచి మీరు ఐసోలేట్ చేస్తే అన్ని స్ట్రెప్టోకోకస్ స్టెఫైలోకోకస్ వస్తుంది, రైట్? సో దిస్ ఈస్ ద కామన్, యూ కాల్ ఇట్ as నార్మల్ ఫ్లోరా. నార్మల్ ఫ్లోరా అంటే మన బాడీ మీద నార్మల్గా బ్యాక్టీరియా ఏవైతే గ్రో అవుతున్నాయో వాటిని మనము నార్మల్ ఫ్లోరా అని అంటాం, రైట్? సో దీస్ బ్యాక్టీరియా ఆర్ ప్రెసెంట్, రైట్? కోకై ఫార్మ్స్, రైట్? అండ్ నెక్స్ట్ యూ హ్యావ్ బ్యాసిల్లస్. Bacillus you have streptobacillus. Streptobacillus means the bacillus which are arranged in the form of a chain, like this, right? This is called as streptobacillus. కానీ ఇక్కడ మనకి staphylococcus అనేది ఉంటుంది, రైట్? Staphylococcus అనేది ఇలా ఉండడానికి ఉంటుంది కదా, ఇలా అటాచ్ అవ్వడానికి ఛాన్స్ ఉంటుందా? ఉండదు, కాబట్టి మనకు staphylococcus ఉంటుంది, రైట్? So you have only streptobacillus and the other form you have Prokaryotic. Prokaryotic means combination of circulate is not neither circular nor rod. Those types are called as Prokaryotic. And next you have vibrioid. We have studied about vibrioid. Vibrioid means comma shaped bacteria. Okay? Yes. And spirillum, and this is last is spirochetes. So you can just see here, right? How the diplobacillus are formed. Student, this is one bacteria. It will be dividing during cell binary fission. Two forms. Avakapothe completely separate avakapothe it forms diplobacillus. If it is multiple divisions and if it is not dividing properly, then it forms streptococcus. Coccus forms, right? Tetraoids. If it is forms two divisions. Two divisions అయినప్పుడు if they are not dividing properly, it forms tetraoids. And this is Staphylococcus. Exact grape bunch and grape bunch laga kaakunda ila bunch మాదిరిగా కనిపిస్తుంది అన్నమాట, right? Those are called as Staphylococcus, right? And square shaped bacteria are also there some. Okay, this. So now let us come into the actual topic, that is ultrastructure of bacteria. Right? So you can just see the structure of bacteria here. This is a prokaryotic unicellular organism. It is a single cell where it functions. All the activities, all the metabolic activities will be carried with a single cell. One cell will perform each and every activity which is necessary for its growth. Okay? So here this is called as unicellular organisms. And if you see their structure, Madhavi, you can just see it is like a chocolate, right? Yes. You are able to see it like a chocolate. Like you can just see eclipse. Eclipse chocolate lo lopala mottam gel laga koncham soft ga untundi and baita mottam hard ga untundi kada, same like that you can just see, right? So here this outer one is called as capsule and inside you will have a cytoplasm. And these bacteria are generally called as prokaryotes, right? Why these are called as prokaryotes? Prokaryotes. Bacteria are prokaryotes. Prokaryotes means they contain primitive nucleus. Primitive nucleus antam. Pro means primitive. Karyotes. Karyotes means nucleus. It contains primitive nucleus. To say example, in clearly you can just say there is no nucleus is present. No nucleus. Only it contains the genetic material, that is DNA is present. The DNA is diffused in the cytoplasm. Right? It contains only the cytoplasm. It doesn't contain any nucleus. Okay? And apart from that, it has some external appendages. The morphology of the bacteria, it contains external appendages. External appendages you can just see, that is a flagella, the outer one, this long hair-like projection, which will be helpful for motility. The bacteria moves by flagella, right? So some bacteria, it is not compulsory that each and every bacteria should have a flagella. Some bacteria will be more motile and some bacteria may not be motile, right? ఏవైతే మూవ్ అవుతున్నాయో వాటిని motile bacteria అంటాము. If they are not able to move, we call them as non-motile bacteria, right? They don't contain any flagella, right? And we will study about what are their arrangements of flagella also in coming classes, right? So now when you can just see the other external appendages, what you can see on the bacteria cell or pili. Pili is these short hair-like projections. You are able to see it, right? Yes. These are called as pili, right? And the outermost layer which surrounds the bacteria and which protects the bacteria from phagocytosis or other pathogens or parasites, which protects the bacteria from invasion, that is called as capsule. Capsule is the outermost protective layer which is present in the bacteria cell, right? And next to this capsule, you will have the cell membrane. Here it is written as reverse, right? And you have cell wall. Next to capsule, you have cell wall, right? Cell wall is generally made up of peptidoglycan layer. Generally, if you take animal cell ki plant cell ki difference entandi, cell wall. In animal cells, you don't have any cell wall. In plants, you have cell wall. The cell wall is made up of cellulose, right? And in fungi, the cell wall is made up of chitin. These are some entrance questions you can just take, right? So cell wall is absent in animal cell, right? Both eukaryotes, plants and animals are also eukaryotes, but in plants you have cell wall and in animals you don't have cell wall. That is a major difference between plant cell and animal cell, right? And next to the cell wall, you have cytoplasmic membrane, or you call it as plasma membrane, right? Plasma membrane will surround the cytoplasm. Cytoplasm means what? Cytoplasm is a liquid-like composition which is present inside the cell, which will protect the cell from osmotic shocks, right? In the cytoplasm only you will have the DNA which is diffused. You can just see this is the cytoplasm. In the cytoplasm, this is the DNA which is present, and apart from DNA, it contains this bacterial cell contains extra chromosomal genetic material or extra chromosomal DNA, which you call it as plasmid, right? And it is not compulsory that every cell should have plasmid. Plasmid undochu, plasmid undakaponochu, right? But because of the plasmid, it may have some special characters because it is a DNA, right? So DNA undadam valla it adds additional advantage to the cell. It has some special characteristics, right? And you have the other ones like teichoic acid is all present in the cell wall itself, and inclusion bodies, right? Inclusion bodies, you'll have some granules, glycogen granules, PHP granules, gas vesicles. Like this, you have some inclusions in the cytoplasm, and apart from that, we have spores formation. Bacteria have a special characteristic, that is formation of spores. If it is not having favorable conditions to grow, if there is no favorable conditions to grow, the bacteria turns itself into a spore, right? And we call them as endospores in case of bacteria. If it is not having favorable conditions like temperature or food or pH or any external parameters are not supporting their growth or which are not helpful for its proper metabolism, then it withdraws all these structures and it becomes a round ball-like structure which you call it as a spore, right? So those are the resistant structures which are dormant structures antam, right? So like this you have these other structures, and when you see the flagella, first structure enti we have studied about flagella kada, long hair-like projection. If you see the structure of flagella, its length is longer than the cell. Cell kante kuda podavu untadi kada, manam kontamandi chustam long hair they have, right? you'll be able to see in TVs, right? If you use so and so hair oil, the hair becomes so longer. It will be longer than your size or your length or your height. So here the flagella is one such hair-like projection which is present in the bacteria, which is an external appendages. It helps in the motility. You can just see here like hair-like projections, right? And here the motility, as I told you, flagella is helpful for motility or movement, and this swarming occurs with some bacteria that is spread across petri dish, this movement, right? And arrangement basis of their classification, as I told you last class, based on the number of flagella, right? As I told you, the bacteria may contain flagella or may not contain flagella, and its number varies. The flagellar number varies from cell to cell. If it doesn't contain any flagella, we call it as atrichous. Okay? And if it contains single flagella, monotrichous, single flagella. Right? If it is contains a tough flagella and a bundle of flagella on one side, it is called as lophotrichous. You can just see it is on the slide also, right? Lophotrichous. Monotrichous, lophotrichous, that is tough of flagella at one end. At one end you have a bundle of flagella, tough and a bundle, right? And next, amphitrichous. Amphitrichous means if the flagella are on either sides, the tough of flagella on either sides, you call it as amphitrichous. What it is? Amphitrichous. Amphitrichous flagella, right? Lophotrichous, amphitrichous, monotrichous, and if the flagella are arranged around the cell mottamlo manaki, if they contain the flagella, these are called as peritrichous. You can just imagine which will be the fast moving bacteria. ఏది fast గా మూవ్ అవ్వగలుగుతుంది? Peritrichous. Very good. E. coli is the best example where it has peritrichous flagella. Around the cell, total around the cell, you have the flagella where it helps in the movement of the bacterium. Right? You can just see this one as we have studied in the last class, hanging drop experiment. Hanging drop experiment is used to observe the motility of the bacteria. Right? And flagellar staining, we have studied about Albert stain. Albert stain is a staining method which is used to stain flagella. Flagella can be identified by flagellar staining. Right? So you can just see these pictures, how the flagella are there. See here. A, B, this is only single cell with one flagella, and this B picture, on either sides you have flagella, and this is on one side, tough of flagella, that is called as lophotrichous. And you can just see this is peritrichous. Right? So these are the different pictures which shows you the arrangement of flagellas. Different flagellar arrangement, and their number is varying from cell to cell. Anni cells lo okate flagella undalani ledu, right? And here how it moves, peritrichous flagella, monotrichous flagella, you can just see. It's like acting like a motor. You'll have the structure actually. You can just go it later on, right? Tuft of flagella will be there, which will be helpful in movement like motor. లోపల మోటార్లో ఉంటుంది కదా, మోటార్తో పాటు అది ఇట్లా విప్ లైక్ మూవ్మెంట్ విల్ బి దేర్, రైట్? అండ్ బేస్డ్ ఆన్ దట్ మూవ్మెంట్ ఇట్ మూవ్స్ ఇన్ forward direction. Like that there will be the movement of the bacteria or rotation movements and so on, right? So if you see the structure of flagella, this is already explained to you, you can just see, right? Idu nenu reverse lo draw chesanu, right? From this way you have to see it. From this way idi cell lopala untundi and this is the outer one, right? So outer part long one is called as filament, and this is the hook which will be holding the motor like structure outer hair like projections, and inside it you will have the rings you call it as outer rings and inner rings, right? And this will be a rod, and this will be outer ring ante cell wall lopala untundi, and this is cell membrane structure, and this is cell wall. So 2 rings will be in cell wall and 2 rings will be in cell membrane, right? So this is the basal body you call it as, and that you call it as a shaft. If you see the flagellar structure, it will be in 2 forms, that is the basal body and shaft anta, which is sitting in the cell you call it as basal body, and which is moving out బయట కనిపించేది మనము shaft అంటాం, right? And the filament or the shaft which is made up of hair like single units which you call it as fimbrine, right? So this is how you can just see. చూడండి అక్కడ outer membrane peptidoglycan layer లో ఎన్ని రింగ్స్ ఉన్నాయి, and cell wall membrane లో ఎన్ని రింగ్స్ ఉన్నాయి అని చూడొచ్చు. Total 4 rings will be there, right? And when you see the next one is pili, right? Pili is the next structure, right?